Hello, my name is Ryan Spillane, and I am studying human biology at North Carolina State University. I will be presenting a review of the effects of thalidomide and thalidomide derivatives on angiogenesis in cancer cells for the Molecular Targets and Cancer Therapeutics Symposium held by the European Organization for Research and Treatment of Cancer, the National Cancer Institute, and the American Association for Cancer Research. My literature review was written for the New England Journal of Medicine, whose audience includes healthcare professionals, researchers, teachers, and members of the general scientific community. The New England Journal of Medicine is recognized as the world's leading medical journal, covering over 150 topics in the areas of clinical medicine, scientific research, healthcare policy, and biomedical ethics. In this presentation, I will provide background information on thalidomide and angiogenesis, discuss how angiogenesis is affected by thalidomide derivatives, explain how these derivatives are used in combination therapy, give an overview of multiple myeloma treatment, and list major clinical risks of thalidomide and its derivatives. I will summarize the main content and end with potential future directions of thalidomide treatment and research. Thalidomide was introduced in the 50s as an over-the-counter sedative that was said to be so safe that you could ingest any amount of it and not be harmed. However, in 1961, thalidomide was removed from the market because it was identified as a teratogen that causes phocomelia. So why does this occur? Scientists discovered that limb growth during fetal development relies on angiogenesis, a process that is initiated by vascular endothelial growth factor. From there, the connection was made that some types of tumors also rely on angiogenesis. Finally, scientists demonstrated that thalidomide inhibits angiogenesis through direct interaction with vascular endothelial growth factor. Like many other drugs, thalidomide has been manipulated into several derivatives which have conserved regions and variable regions. In the figure, we can see that the isoindole aromatic ring system is highly conserved and responsible for similar properties. The variable regions are the functional groups branching from the ring, which are responsible for different biological mechanisms. Early research on thalidomide derivatives showed that thalidomide and EM12 have similar anti-angiogenic effects, which is supported by the fact that their functional groups are the same. Later, a new derivative was developed called S3-APG. In one study, this drug was also demonstrated to inhibit angiogenesis, this time affecting a wider range of cell lines and with more intensity than thalidomide, as shown in this figure from the study. More recently, thalidomide was developed into two derivatives that are used in cancer treatment today, called lenalidomide and pomalidomide. Thalidomide is becoming less popular as a cancer treatment because it is associated with more adverse events than other available options. Thalidomide and its derivatives are almost exclusively used in combination with other drugs, including mainly anti-inflammatory drugs and sometimes drugs that trigger regulatory mechanisms leading to apoptosis. One study demonstrated the effectiveness of thalidomide in combination with an anti-inflammatory drug called Solandac by measuring tumor volume after treatment with thalidomide alone, Solandac alone, or thalidomide and Solandac together. Results showed that smallest tumor volume was observed after the combination treatment as seen in this graph. Thalidomide, lenalidomide, and pomalidomide are most commonly used to treat multiple myeloma. Studies show that thalidomide and lenalidomide are similarly effective, but there are more safety concerns over thalidomide. Sometimes lenalidomide fails to successfully treat multiple myeloma, and patients develop relapsed or refractory multiple myeloma. In this case, pomalidomide has been shown to be effective. One study demonstrated that a second treatment of lenalidomide in combination with ixazomib and dexamethasone is also effective. Some of the major clinical risks associated with thalidomide, lenalidomide, and pomalidomide are occurrence of peripheral neuropathy, thromboembolism, and infection. Thalidomide is associated with the highest occurrence of peripheral neuropathy. Thalidomide and lenalidomide are similarly associated with thromboembolism, and pomalidomide is associated with the highest occurrence of infection, although each of these adverse events occur in studies with all three drugs. Clinical trials always include a section discussing adverse events, as shown in this table from a report on a study of the effectiveness of lenalidomide for multiple myeloma treatment. To summarize, we know that thalidomide inhibits angiogenesis. Thalidomide derivatives have different biological mechanisms as a result of variable functional groups. thalidomide in combination therapy is associated with positive outcomes for cancer patients, and thalidomide, lenalidomide, and pomalidomide are current treatment options for multiple myeloma, but are associated with clinical risks of peripheral neuropathy, thromboembolism, and infection. The future of thalidomide will be driven by research questions and focused on minimizing the associated clinical risks. Are the risks directly caused by thalidomide, or are other factors involved? Once that is determined, what methods can we use to reduce adverse events? Can we use different combinations of drugs for safer and more effective treatment? Would it be beneficial to develop new derivatives of thalidomide? Can we use thalidomide and its derivatives to treat other types of cancer? These are just a few examples. As our understanding of thalidomide's mechanism of action increases, so will our ability to develop safe and effective treatments for cancer. Thank you.